Hello everyone, welcome back to Anatomy Weekly. In our last class, we discussed about the skeletal muscles. We are going to continue with the topic of muscles today, and we are going to talk about the cardiac muscles and the smooth muscles. First, let us talk about the cardiac muscles. The cardiac muscle has the same type and arrangement of contractile filaments as the skeletal muscles. Therefore, cardiac muscle cells and the fibers they form exhibit cross striations, which is evident in routine histological sections. In addition, cardiac muscle fibers exhibit densely stained crossbands, which are called intercalated discs. These intercalated discs represent highly specialized attachment sites between the adjacent cells. This linear cell-to-cell attachment of the cardiac muscle cells result in the formation of fibers of variable lengths. Some cardiac muscle cells in a fiber may join with two or more cells through intercalated discs, therefore creating a branched fiber. The central location of the nucleus in cardiac muscle cells is one feature that helps distinguish them from the multinucleated skeletal muscle fibers, whose nuclei lie immediately under the plasma membrane. The intercalated disc represents the attachment site between cardiac muscle cells. In the light microscope, the disc appears as a densely staining linear structure that is oriented transverse to the muscle fibers. Often, it consists of short segments arranged in step-like fashion. The smooth endoplasmic reticulum in the cardiac muscle cells is organized into a single network along the sarcomere, extending from Z line to Z line. The smooth endoplasmic reticulum of the cardiac muscles is not as well organized as that of the skeletal muscles. The T-tubules in cardiac muscle penetrate into the myofilament bundles at the level of the Z line between the ends of the smooth endoplasmic reticular network. Thus, there is only one T-tubule per sarcomere in cardiac muscles. If you recall, the same structures in the skeletal muscles would form a triad. The intrinsic spontaneous contraction or the beat of the cardiac muscles is evident in embryonic cardiac muscle cells as well as in the cardiac muscle cells in tissue culture. The heartbeat is initiated, locally regulated, and coordinated by specialized, modified cardiac muscle cells called cardiac conducting cells. These cells are organized into nodes and highly specialized conducting fibers called Purkinje fibers that generate and rapidly transmit the contractile impulse to various parts of the myocardium in a precise sequence. A localized injury to cardiac muscle tissue that results in the death of cells is repaired by replacement with fibrous connective tissue. Consequently, cardiac function is lost at the site of injury. This pattern of injury and repair is seen in non-fatal myocardial infarction. In the past, it was thought that once cardiac muscle cells are destroyed, they cannot be replaced by new muscle cells. However, recent studies of hearts which are removed from individuals that have received transplants reveal the nuclei undergoing mitosis.
The smooth muscle generally occurs as bundles or sheets of elongated fusiform cells with finely tapered ends. The cells, also called fibers, range in length from 20 micrometer in the walls of the small blood vessels to about 200 micrometer in the wall of the intestine. They may be as large as 500 micrometer in the wall of the uterus during pregnancy. The smooth muscle cells are interconnected by gap junctions, the specialized communication between the cells.
Smooth muscle cytoplasm stains rather evenly with eosin in routine H&E preparation because of the concentrations of actin and myosin that these cells contain. The nuclei of smooth muscle cells are located in the center of the cell and often have a corkscrew appearance in longitudinal section. Smooth muscle cells possess a contractile apparatus of thin and thick filaments and a cytoskeleton of desmin and vimentin intermediate filaments. The remaining sarcoplasm is filled with thin filaments that form a part of the contractile apparatus. Thick myosin filaments are scattered throughout the sarcoplasm of a smooth muscle cell. The thin filaments in a smooth muscle cell are attached to cytoplasmic densities or dense bodies that are visible among the filaments. These structures are distributed throughout the sarcoplasm in a network of intermediate filaments. The components of the contractile apparatus in smooth muscle cells are the following. The thin filaments contain actin, the smooth muscle isoform of tropomyosin, and two smooth muscle specific proteins, caldesmon and calponin. No troponin is associated with smooth muscle tropomyosin. Thick filaments contain myosin II, which differs slightly from those found in the skeletal muscles. The side polar myosin filament has no central bare zone, but instead has asymmetrically tapered bare ends. This organization maximizes the interaction between the thick and the thin filaments, allowing the overlapped thin filaments to be pulled over the entire length of the thick filaments. The dense bodies contain a variety of attachment plaque proteins, including actinin, which anchors both the thin filaments and the intermediate filaments either directly or indirectly to the sarcolemma. They play an important role in transmitting contractile forces generated inside the cell to the cell surface, altering the cell's shape.
Now that we have talked about the three different types of muscles, let us summarize the differences. If we talk about the muscle cell, in skeletal muscles, the muscle cell is large and elongated. In cardiac muscles, it is short and narrow, while in smooth muscles, the cells are short and fusiform. Talking about the location, skeletal muscles are found in the muscles of the skeleton. Cardiac muscles are found in the heart as well as in the superior and the inferior vena cava and the pulmonary veins. Smooth muscles are found in the vessels, different organs, and viscera. If we talk about the fibers, the skeletal muscle fibers are a single skeletal muscle cells. The cardiac muscle fibers are linear branched arrangement of several cardiac muscle cells, while in smooth muscles, the muscle fibers are single smooth muscle cells. If we observe the striations, in skeletal muscles, striations are present. In cardiac muscles, striations are present, but in smooth muscles, striations are absent. The nucleus in the skeletal muscles remains peripherally. In the cardiac muscles, the nucleus remains centrally, and in the smooth muscles also, the nucleus remains in a central position. If we talk about the T tubules, very prominent T tubules are present in skeletal muscles forming a triad. In cardiac muscles, the T tubules are present at the Z lines forming a dyad, while in smooth muscles, we do not observe any arrangement of T tubules. In skeletal muscles, there is no cell-to-cell junctions. In cardiac muscles, we find a prominent and distinct intercellular junction in the form of the intercalated disc. In smooth muscles, we find gap junctions. A specialized feature of the skeletal muscle that distinguishes it from the other muscles is the well-developed smooth endoplasmic reticulum and T tubules. In cardiac muscles, the specialized feature that distinguishes it from the other muscles is the intercalated disc, while in the smooth muscles, the specialized feature is the presence of dense bodies. Let's talk about the way these muscles function. The type of innervation of the skeletal muscles is voluntary. In cardiac muscles, the type of innervation is involuntary. In smooth muscles also, the type of innervation is involuntary. Talking about the growth and regeneration of the muscle cells, no mitosis is observed in the skeletal muscle cells. Usually no mitosis is observed in the cardiac muscle cells too, but frequent mitosis and regeneration is observed in the smooth muscle cells. In response to increased demand or overuse, the skeletal muscles undergo hypertrophy, the cardiac muscles too undergo hypertrophy, while the smooth muscle cells undergo both hypertrophy as well as hyperplasia. So that's all we have for you regarding muscles. Stay tuned to Anatomy Weekly. Keep watching, keep learning. We'll see you next time. Thank you.